So just a short outline. We will start by introducing surface plasmon resonance, which we assume is familiar to most of the people listening, and we will specifically focus on the proteome array format. Next, we will just introduce the topic of protein engineering and selection, since that's our main technology. We will talk a bit about small protein scaffolds, how to select proteins from combinatorial protein libraries, and specifically introduce the albumin-binding domain, which is the scaffold we mainly use. And then we will be more experimental and talk about bispecific binding proteins, which we have developed based on this albumin-binding domain or ABD scaffold, go through some published examples and also some unpublished recent data, where we mainly focus on binders to cancer targets. Alright, so the surface plasmon resonance platforms, we guess most of you are familiar with. I will not mention everything which is said on the slide, but as you all know, it's used to detect binding events on the ligand immobilized on the surface, and it's a very useful technique for studying of interactions, and in our case, mainly protein-protein interactions. So a binding event on the surface results in a signal which can be coupled to the interaction. The definition, I won't go through everything, but there is a physical phenomenon happening upon binding, more or less related to the amount of mass bound to the surface, and this occurs as a thin layer, and then this can be used to derive kinetic data, equilibrium data, the active concentration of an analyte, and many binding parameters. It can also be used in competition format to look at how different analytes compete for targets, for example. We have some examples later on. An SPR system, for example, the Proteon XPR36, consists of fluidics, optics, sensor sheets, controllers, and software. In our case, we mainly use anion coupling of ligands, so the sensor sheets are normally DLC sheets and sometimes GNM sheets. And the data can also be evaluated in the program, and you will see in some of the pictures how we evaluate data and fit curves and calculate kinetics. So just to remind you all that to work on SPR, you need a surface, which is normally a polymer with functional groups that can be used to couple ligands, in this case illustrated by an antibody, and you can monitor the binding of an analyte which is flowed over the surface. And of course you can alter the coupling chemistry, but as I said, we will mainly talk about anion coupling. And the power of label-free analysis, as you all know, is that you don't need to label and you get the kinetic data in one experiment. So you can, for example, look at how fast an interaction takes place, how stable the complex, the AD complex in this formula is, how strong it is if you look at growth rate constant, and you can also look at specificity and watch binding to other related or unrelated proteins. And you can use this for plenty of biomolecular interactions. We work mostly on protein-protein interactions, but you can also look at peptides or small molecules, DNA, carbohydrates, lipids, etc. But the focus here will be on protein-protein interactions. The data are plotted in sensor graphs, which is represented here up at the top, where you have real-time measurements and you follow the binding to the surface, which is measured in response units. And this can be seen as the mass bound on the surface, more or less. The protein uses something which is called the Kretschmann configuration, where the ligand is coupled in a channel on top of a gold layer, and through light and the reflected light in a prism, the amount of found analytes can be detected. So here are some slides that we got from Bio-Rad, where we go through the experimental steps, which are probably familiar to most of you. So you start with a surface coated with a layer suitable for coupling. You activate the groups in the first step, and in the next step, ligand is immobilized. Through anion coupling, usually it could also be captured, for example, and the unreacted groups are next deactivated. To perform the analysis, an analyte is flown over, and you monitor in real time the binding of the analyte to the immobilized ligand, and you can get data on how fast it binds and when it reaches equilibrium, etc. And this is then used to derive kinetic information about this specific interaction. When you remove the analyte from the flow, it starts to dissociate, so you can also follow the dissociation, and from the rate of association and dissociation you can calculate the kinetics. And to prepare the surface for a next injection, it needs to be regenerated, so you inject something that gets the remaining analyte to come off the ligand. And this is probably familiar to most of you, but for example, you watch the analyte A binding to ligand B, forming the complex AB, which is described by the rate constant, the association rate constant, which explains how or determines how fast this interaction takes place, and the dissociation rate constant, which is related to the stability of the complex. You can also measure the affinity, which is often expressed as a dissociation equilibrium constant, or KD, or an association rate constant, which is the inverse of the KD. And if you look at conservation of mass and the steady state, you can see how this dissociation rate constant can be calculated if you assume that the amount of formed complex is it doesn't change over time. You can get the equilibrium state and calculate the dissociation rate constant. So normally if you talk about KD values, a low number, for example a nanomolar, would describe a strong interaction, whereas a high number is not that strong interaction. And as we said already, looking at one of those sensograms, you start off from a baseline, you follow the association, and during the association both binding and dissociation takes place. When you stop injecting the analyte, you will monitor the dissociation, and you can mathematically explain those different processes and use it to calculate the kinetics of the interaction. And just to remind you all that the kinetics is important, in this graph is 4 different interactions that have different rate constants, but the same KD value, the same dissociation constant, the same equilibrium constant. So looking, for example, at the very fast binders that comes off very quickly, may be good for one application. For example, a sleeping pill, which should hit quickly, but the effect should not stay for too long. Whereas the others, where you have a binding that isn't as quick, but the effect lasts for a longer time, maybe, for example, a painkiller. So kinetics is an important part of all interactions, and you should perhaps be a bit careful about only talking about the equilibrium constant. Okay, to sum up, the basic SPR technology advantages is that you don't need to label the sample, you don't need to label the ligand or the analyte. You can monitor the interaction in real time. The ligand can be reused if you find proper regeneration conditions. You don't consume very much sample compared to other techniques, for example, ELISA, and you also get kinetics. You get the full on and off rate profile. It can be automated, and especially on the protein, you can run samples at a rather high throughput. So let's focus now on the Proteon system, which is an array format having a Criss-cross fluidics design, which is very useful, we think, for different assays. So on this instrument, you look at different spots that have either been immobilized only with ligand or only met analyte, or what is most interesting, been immobilized with a ligand, followed by injection of an analyte, so you get to measure the kinetics. So it's a perpendicular array where you have 6 channels in one horizontal axis and 6 vertical channels, which allows simultaneous monitoring of 36 interaction spots where those channels move. And this leads to reduced experimental time, higher throughput, and since you have spots that only met ligand or analyte, this can also be used for real-time referencing. And now we'll show a small animation to clarify what we mean. So So if you look at the censorship surface, first you have this multi-channel module which decides which direction the flow on the channel should run. You start by pressing it down on the chip in one direction, and it can be lifted up and switched 90 degrees. So if we look at an immobilization, for example, the first step will be to activate the channel. And in this case, it's a typical amide coupling. So you use NHF-EDC chemistry to activate the surface. Next, the ligand is injected in the same direction. In this case, illustrated by 6 different antibodies injected in one channel each. And then deactivated to cap the spots that weren't coupled with the ligand. So now we prepare the surface containing 6 different ligands. And by changing channels and rotating this NCM, you can now inject an analyte and in real time follow the binding of this analyte to all the 6 ligands. So this could be 6 different analytes, or it could be 6 concentrations of the same analyte. There's a lot of flexibility. And when you stop injecting the analyte, it starts to dissociate, and you can monitor the dissociation. So here's an example of how it may look. You inject the sample over those 6 different surfaces, and the reference shown to the right. You can follow the association when you stop injecting ligand and bring the dissociation phase takes part. And in real time, you get a lot of data. So in this case, you get binding profile for 6 different concentrations over 5 different ligands, and you can compare and reference against the blank surface or an unrelated protein if you would prefer. Alright, so that's it about the SPR background, and I think most of you have heard this many times before. So now we will switch into our focus area, which is protein engineering and library. So if you have any questions, just let me know or I'll continue. Alright, so let's move into protein engineering. Protein engineering is the main thing that we work on here, and the aim is to modify existing proteins and to some extent provide new proteins never seen by nature for molecular recognition. You may also have an existing protein which you want to improve for stability, solubility, activity, or if you have something that doesn't get well expressed in bacterial hosts, you may also try to alter the expression levels by engineering the protein. So in the toolbox we have 20 amino acid building blocks, and by making different mutations either in a rational sense or using combinatorial methods, you may select for improved variants. So in our case, we focus on molecular recognition. We try to engineer existing proteins for improved molecular recognition or introduce novel binding sites on protein domains. And as you probably all know, the use of affinity proteins, they have many different application areas, and mainly people think of antibodies as the affinity proteins. So, for example, antibodies or other proteins are very useful in basic research. They can be used for purification or detection, in diagnostics, and sometimes in therapy. Antibodies, as I said, are the main affinity proteins. They are historically derived by immunization of animals, which is a process where you have a limited control. It's hard to modify. There may be differences between different animals. The antibodies you receive in the end are normally polyclonal, which means they have hopefully the same binding, they recognize the same antigen or target protein, but they are different molecular species having different kinetic characteristics, for example. Since it's an animal origin, this may also be a problem if you use them for in vivo applications in man, since you may get an immune reaction against the animal compounds. And another limitation using immunization techniques is that you cannot generate an antibody binding to a protein which is recognized as self, since you shouldn't be reactive against self proteins. Or if you have a target or an antigen which may be toxic, this will also cause a problem if you try to inject it in the host to use antibodies. And as you all know, the antibody consists of a constant part where effector functions reside in this fragment crystallizable. It's also the reason antibodies have a long half-life. There are receptors binding this F2 part that recirculate antibodies and keep them for a longer time in the blood. And each antibody also has two antigen binding regions formed by variable domains of the heavy and light chain. Since the beginning of antibody engineering, a lot of things have happened. There is just some examples. For example, now antibodies can be generated in vitro by hybridoma technology, where cells from a mouse are fused with cancer cells to provide the source for monoclonal antibodies, which can be reused. Antibodies can also be produced by recombinant means. They may be humanized. If you have the gene for the antibody, it can be combined with human genes, so that the part of the antibody having an animal origin is smaller. And now it's also possible to get antibodies from transgenic mice carrying genes from humans. And of course, more antibody fragments as well. Alright, so let's talk about combinatorial libraries, which is the focus of our work. So the main idea is that you start from a protein, in this case a small protein domain shown to the left. You identify positions in this domain that you would like to target for mutagenesis. And then you do this mutagenesis by some laboratory means, and this provides something we call a library, which is a large pool of different variants represented by all the smaller proteins in the middle. And then you would like to select a member of the library having some desired property. And in most cases, this is binding to a target protein. So the target protein is illustrated by this thing on the hook. And then you need some system to select the binders from the background of non-binders. So the parts in the system are the library, the target protein, and some kind of system to select the molecules you desire from the pool of many different variants. This is just another way of showing it. You start off from a combinatorial library. You implement some kind of selection process in order to select variants having desired properties for your application of choice. There are many different kinds of libraries. As we said before, they may be immunized, which means they are originally derived from an immunized animal. It could also be a synthetic library, which is produced by some rational design in vitro, and it has never met an animal. Libraries may also be naive, which means that they are not directed against a particular antigen. They are totally random, so to speak, so that they may be selected against any protein. Or they may be targeted, where you have some previous knowledge which you implement in the library design, and hopefully the library will be enriched for proteins binding or specific target or having an increased stability or whatever your aim is. The randomization or diversification is normally implemented on the genetic level. So at the bottom is a representation of the gene for this protein domain, and by putting in the generous codons at specific positions labeled here by the dots, you get the variability that represents the diversity in the library. And NNK, for example, is a codon which encodes 32 different codons. M can be any of the 4 bases, and K can be 2 of them. So this yields, for each position, all the 20 possible amino acids as well as a stop codon. So you can just imagine the diversity if you implement this kind of codons at many positions at the same time. Here's an example of the typical workflow we use. So we start off with a combinatorial library and use some kind of selection system. Here it's represented by a stage carrying the protein of interest. We will go through this a bit more in detail in a while. Next, after the selection, we do some kind of initial screening. It may be by sequencing or by ELISA or some more functional test. Interesting candidates are expressed and purified, and in the end, we would like to see whether they bind the targets we intend to get binders to, and here SPR is a very useful technique to study this. So now we will move from antibodies to smaller protein scaffolds. In the box to the right are some dimensions for an antibody, and as you can see, it's a rather big molecule. There are dimensions on the box in nanometers, and the small domain which we focus on, we will tell you a bit more about, is called an albumin-binding domain. It's about 20 the size, or even more, of the antibody, and the dimensions are much smaller. So there are many advantages of using smaller binding proteins instead of antibodies. One thing is that they are more easily produced. You can more easily produce them using recombinant methods and produce them in bacteria or even by synthesis. Normally, if you choose a scaffold for this purpose, you normally choose something that doesn't contain cysteine, since you don't want to get dimerization and limited control due to formation of disulfide bonds. Since the smaller proteins lack the Fc part of the antibody, they don't induce any effector function if you will apply them in vivo. They are often thermal and chemically stable, and of course you may choose a domain or a protein as your scaffold which has those advantageous properties. Normally the folding process is also easier for smaller protein domains, and if you apply them in vivo, you may get a better tissue penetration and faster clearance compared to an antibody. And this may, for example, be useful if you have something binding a cancer target, as in our case, and you would like to image it compared to background. So you inject the binder, and over time it accumulates at the tumor, and what is not bound to the tumor will quickly get secreted from the body, and you will get some kind of contrast in the targeting. So here is just an example of the domain we work on. It's called the albumin-binding domain. It's a small domain, only 5 or 6 kilodaltons, contains 46 amino acids, and it has got an inherent binding to human serum albumin. So HSA is human serum albumin. And we have incorporated, we have used it as a scaffold for a library by using mutagenesis to target the opposite side of the molecule to provide a new engineered binding surface. So we have a small video here that will show how this looks. So this is the small molecule, a space-field representation. It goes into a 3-helical bundle, where those green areas that just quickly flashed by are the positions that we target for randomization, and the blue are the ones responsible for binding to albumin, which we do not change in the library. So the outcome is a small domain where one side binds to a target of choice, if we're lucky during the selection process, and the other one binds to human serum albumin. So 2 binding sites implemented in a small domain. And talking about the selection systems, if you remember the fishing rod in the previous slide, there are many different ways of selecting your candidate binder from a library. For example, you can use systems that depend on cells, which is our focus. So here represented by phage display to the far left, or cell surface display. So the main idea is that every candidate of the library is expressed on a cell or a phage particle and physically linked to the gene encoding the same library member. So by mixing the big population of phage with your target protein, normally immobilized to solid support, you may select for proteins that are binding, and at the same time you select the genetic information providing the characteristics of this particular library member. So here's an example of how this works. If you consider a phage display, you have an immobilized target, the turquoise one, and you mix it with your unsorted phage display library. So you have many different variants, Each column represents the variant in your library, and each one is carried by the same phage particle, but it also carries the genetic information. You mix your phage library with your target in a selection. You wash away the ones that don't show any interaction, and then you elute, normally by changing the pH, the ones that are bound. You use this eluate to infect bacteria, and the bacteria are used to amplify the selected phage, which will start the next round of selection. So here are some more specific details about how we use those systems. So as we said before, we use the small albumin-binding domain, which only includes 46 amino acids. We've identified 11 residues exposed on the surface of 2 of the helices, and we've targeted them for randomization. We produced a library which is expressed on phage. So in the first example, we use this library to select binders against the protein instead. And in this case, since we don't touch the albumin-binding side of the molecule, which resides, if you remember the movie, resides in the second helix mainly, we can use this protein that is selected as a tag and couple it to a target protein to be used for purification of either a column carrying the cell domain, which we select binders for using this new randomized surface, or we can utilize the inherent binding to albumin for purification on another kind of column having immobilized albumin. So in this case, the resulting protein we get out from the strategy is useful as a tag for purification of proteins. In another example that we will just very briefly mention, we use the same technique to select binders to TNF-alpha. We had a look at the outcome from the phage display selection, used this to make an affinity maturation, and in the end we have binders binding to albumin and TNF-alpha, which may be useful for treatment of patients having autoimmune inflammation. Alright, so that's the first example once more. The binding protein, we did a selection, we managed to identify one molecule having affinity for the cell domain, which was the aim of the selection strategy, and it also has retained affinity to albumin, and those dissociation equilibrium constants were determined by SPR. By fusing this protein, represented by this yellow box, to different target proteins having different molecular weights and different charge, different isoelectric points, we show that we can use this tag for specific purification either on a column immobilized with human serum albumin, followed by a column immobilized with the cell domain, which is the other target. So through the two specificities inherent in this small protein domain, we can actually utilize it for purification of either an albumin column or a column carrying the other target protein. And here are just some chromatograms and phage scales to show how you load the bacterial lysate where you overexpress one of the proteins, either over the small domain itself, or in one of the medium-sized proteins or the larger protein. And by passing it through an albumin-containing column, you get a much purer product, but by utilizing the second affinity, you can increase the purity even further. And at the bottom is only the same thing, shown having the step with the cell affinity shown first, followed by the albumin, and you get the same result. So the tag itself is emitted in this bottom picture. And as the last example of what we've done is to use binders selected using phage display to TNF-alpha. You can also do a new library based on the first binders you identify. And in this case, we have used flow cytometry and expressed this library on the surface of cells, cephalococcal cells. And using fluorescently labeled TNF-alpha or albumin, we can select populations of cells, which means different variants of the library that interact with both or one of those target proteins. So it may look something like this, that you sequence the area, we target 11 positions for randomization. So here is the sequence of the domain from the beginning, 46 amino acids. The boxes denote the location of the helices in the structure. 11 positions are indicated by dots that were targeted for randomization. And then we came up by sequencing close after the selection. We managed to identify 2 that were shown by XPR to bind the target, and then we can sort the full library expressed on the staphylococcus and enrich for binders. So those are just dot plots from a flow cytometer showing target binding versus expression level. And the more cycles we run in the flow cytometer, the more enriched is the population that is expressed well and is binding the target. So this is also a way of sorting the combinatorial libraries. Now we'll focus more on cancer targeting, which is the main topic, and now it will become more experimental. So I will leave the word to Mikael, who will continue. And if you have any questions, you're free to join.
Okay, thank you, Johan. So we will now move over to an ongoing project. There's no published, so we will not disclose too much information. So just to give you just a quick summary of what you understood, just to emphasize a few points. So the 2 key features we have with the domain is number 1, we have molecular targeting to directed evolution, and this is achieved by the surface that we have engineered on the opposite side of the ABD or the assay binding surface. And also the second feature is utilized through the HSA interaction, and what this enables us is longer in vivo half-life extension if we would use this for in vivo applications later on. So the way this works is we have an increased hydrodynamic gradient for associated with albumin. So that is the pathway you are using, and this also enables us to take advantage of recycling through the neonatal receptor, which has been proven very successful by doing all sorts of ABD fusions or HSA fusions. So the workflow or the summary of our current project, what we've done here is we have first used base display selection against the target we are interested in. We have identified different binders and we characterize them. And then finally we have done a composition study to evaluate the epitopes that our candidates are binding to. So let's dive in, and we have used XPR for all of this. And why have we done that? It's because it's a very rapid, reliable analysis of protein interactions, as you may well be aware of. It's label-free, it's real-time, and it requires pure protein. That could be a negative aspect of it, of course, but we have a very, very robust method to purify our proteins. We're utilizing the HSA binding feature, and we get very, very pure protein with this effort. So what we need to do is we have 7 candidate proteins in this example, and we have 2 targets. So we have a base target, and we have our newly engineered binding feature on the opposite side. So this gives us 14 interactions in total, which is quite a lot to do on SPR. And so that's why we're taking advantage of the high throughput capabilities of the Proteon and the ability to make parallel interactions. And also we want to do competition experiments to see if we can compete with the natural growth factors. That would be interesting to find out for functional purposes. See if we can use these later on down the line. And also we define analysis in triplicates to really be able to trust our data, and that's why we go to the protein. So a typical setup that we use is quite straightforward. You have 6 surfaces. We have used 5 of these to bind to immobilize a target. These could be different targets, and we have different candidate molecules. And also we have 1 blank surface. And for our analyze, we chose to inject an array of different concentration, so we go from high to low, and then we also have a zero concentration for referencing. So the blank and the zero injection enables us to do double referencing to really have reliable data in the end. Challenges that occurs when you do this is, first of all, normalization. It's hard to find a good pH dependent on your TI of your protein to enable very effective normalization. The pH used can be predicted through the TI, so that's where it can be helpful. But the regeneration is a little bit more problematic. If you regenerate too harsh, you will ruin your protein over time. If you have too weak regeneration, you will have no regeneration at all. And this requires a little trial and error. You have to sort of see what works. You have to try this with something, try something first, and then if that doesn't work, you have to work your way forward. And of course, with all of this, it's also challenging to get good data, but that's how it is with everything. And in our case, we always need kinetic data. We need to evaluate the KD, the dissociation equilibrium constant. So that really requires us to have a lot of data to base that on. So here's just an example where we can go from bad data to good data. And as you can see on the left, we have very, very low response, total response, and we have very blurry signal, and it's really hard to make anything out of it. We can barely see that it's binding. And in this case, we use pH 4.5 for mobilization, and we use assays for regeneration. So this apparently was not very good for this protein. We could observe immobilization, so that was not the main problem, but when we changed to pH 5, we can see more effective immobilization, that's one thing. But also we changed to regeneration, and then we went from seeing lower and lower max response in our analytes to getting very, very high and very stable max responses to get very comparable between each analyte injection. That's very nice to see, but that requires some optimization, and that's always time-consuming, but it was worth it in the end. So here's just some results from this, what you can do. So here in this example, we have taken two surfaces, and we have varied the amount of protein that we have mobilized. And this gives us different max responses, as you can see. But the data is quite reliable on all surfaces, and we get good overlaps when we do the evaluation, the kinetic evaluation in the software. So at the bottom you can see the data you got from this particular example, and those results between surfaces overlap quite well. In this example, we see some artifacts that we get that the KD is actually affected, is actually dropping with each surface. So that could be an artifact in this case. The variable immobilization level can also be used for optimization in this case, where you can actually find a good immobilization level for further analysis. So that's actually something we use for optimization. But in this case, we've got quite reliable data. And here's another example where we have evaluated the HSA binding surface of these molecules. And we have also included MSA here in the two surfaces down here at the bottom, the number 5 and 6, and number 4 is used for reference. So in this case, we have very nice response levels. They're up at 200 in most of the cases, and for the two lowest mobilization levels here, for HSA, we get much lower, but we get still very reliable data. And what can be commented about this here is, as you can see, we have very, very slow off-rates. So the KD values are up at 10 to the power minus 5, which is kind of close to the limit of detection. So one comment you can have about this is that we should probably monitor this during a longer time. So that's one problem you can have, and that's something you can't really know beforehand. So this is also an issue that needs to be addressed, but they can also, you can just do another injection and you have the problem solved. So that's a very nice example. You see that the kinetic data is actually quite similar between the surfaces, so you can, in this injection, easily use, you can calculate the standard deviations and use that for publishing, so that's very nice in this case. So what we have is we make two selections, again, two quite similar targets, and we try to evaluate those binders. And the end result is that we have several candidates, and one group of them binds its respective target, and the HSA binding is retained compared to the original scaffold. And the other one, we can see the same thing. We have affinity for its target protein, but not the other, and we have retained HSA affinity. So that easily showed us that it's possible to engineer a bispecificity into single domain. So relatively quickly, I would say. Okay. So further on, we wanted to evaluate the epitopes that the binders bind to. So we wanted to check if they can compete with the natural growth factor that binds to the target. So this setup, we use growth factor that we immobilize on the surface, and we inject the target with or without our analyte. So on the left, you will see a typical reference injection. So you have just the growth factor and you inject the target. And then in the actual sample analyte, when you sample injection, you have ABD, and you can vary that, as we will see in the next slide. So in this example here, you will have one ligand and two analytes injected simultaneously. So this will not be co-inject, unless it will just be pre-incubation. Okay, so here's to show you how it looks like. So we have growth factor, you have that immobilized on 5 surfaces and have a blank. So this kind of enables you to be creative. You can, if you have more complex systems, or as we are working on it currently, where we want to look at if they can interact with each other as well, you can just use these different ligands on the surface. But here we have just taken 5. And this will enable us to do the analysis in many different directions. So that's very nice. So you inject the target, and you vary the analyte concentration, as I said, and you have a 0 injection there as well. Important to note is that we also use an injection where we have only targets, the same target concentration in all the channels, and that we will use for normalization, as I will show you in a minute. So here's some examples of the output you can get to this experiment. So at the top you have the reference injection, and as you can see, this is the same concentration in all the channels, but it will still have some small variance. So that was expected because the mobilization is probably not perfect throughout the entire channel. It should be quite an even distribution, but you never know. So that's taken into account here. And then we can move on to the second panel. We have the sample, and this is a good example of what it may look like. So this will be referenced afterwards. So this is not referenced directly. This is what you will get out in the raw data, and you will see even now without referencing that you have a distribution where we go from the lowest concentration, or actually the 0 concentration of analytes and just the target, and then you have higher and higher concentration as we go down. So you can immediately see in raw data that it's actually working, that you can actually interrupt the binding of the target to the surface. And at the bottom, we see a positive control, very, very high affinity binder, and you can immediately see that as soon as we reach the second concentration, we have reached kind of a maximum inhibition. And so it's very, very clear in the raw data. But as I said, we will do referencing towards reference channel, and the way we do this is that we extract data from a particular time point, and this will have to be done manually. It can be done in the software, but then we export it to, for example, Excel, and then you do referencing there. So the workflow is mobilization. You have to check the surface functionality with reference injection. Then you inject your panel of analytes, and this is quite time-consuming here because, as I said, it's a pre-incubation. So we actually did 1 hour pre-incubation, which is quite long. So imagine doing 1 analysis, it takes you 1 hour, and if you are effective, you can maybe start several pre-incubations at a time, or with a certain delay, and you can get you can get the data quite easily that way. So then you extract representative data points, as I said, and you have to, of course, take the data points at the same time, and you normalize them to reference manually. So one thing to note here is that one trouble that we encountered is that when we started doing injections right after immobilization, we got quite low responses. And we didn't really know why that was. So we decided that we will leave it in standby for overnight to see if we can, if the coating on the surface is not really folding properly, if we could make it better in some way. So we did the same kind of reference injections the next day, and immediately we see a very, very high increase in match response. So that was very nice to see. So, but this could of course vary from case to case. In this case, the protein is very small that we are mobilizing, about 7 kilodaltons, so it's quite small, and we don't really know much information about the fold, so that could be something to take into account. But this works very well to do all analysis, and then also at the end we do another reference injection just to see that it's not, they don't have any bias. So this is the result we get from this. So just to quickly show you here, so then here at the top you have a negative control, first of all. So this is an ABD in this case that is not associated with target in any way. So as you can see, it's kind of, it doesn't really interrupt binding with increased concentration. So just to note here is that in each data point here, you actually have this is the mean value of 5 because we have 5 surfaces. So that's just to show you that it did very, very, a lot of replicants in heavy injection. So you get very reliable data. And then as you go down here, these are ABD molecules that are being injected here. So we have 4 of them here. So the red, green, the dark blue, and the purple. And they actually line up, or as you can see, they are leveling out at different levels on the y-axis. And these are actually lining up in order of affinity. So the higher affinity, the lower you get on the y-axis. And at the bottom here, you have some positive control. You have a very, very high affinity binder here in orange. And in light blue here, you have actually the growth factor inhibiting itself. So the binding is cancelled that way. But it's interesting that by increasing the concentration, you can't really affect, you can't cancel it out completely, which is interesting. This is hard to explain, but we tried also with an ABD here to increase the concentration even further and see if we could cancel out more, but that was not the case. So clearly this is something, this is an application specific for this assay, and it could be further evaluated with a different target on the surface or something like that. But the results are very nice, and clearly you can very reliably say that our candidates are actually binding to this epitope and binding to probably the same epitope, since they are very similar in sequence. So related to this is you can actually use another application, or there are many applications you can still use this one application example. So we want to know, can we for simultaneous binding? So can our analyte find the target and, for example, or HSA in this case, simultaneously? So in this example, we have HSA on the surface, and in the same way as before, you inject the analyte, which was the target in the last experiment. We inject the analyte in the same concentration, and we vary the target instead. And you use this in the same way, you do referencing in the same way, you can do reference against blank and the zero injection. This is the same way as before. And this is the example of the raw data you can get. You have a reference surface, very nice in this case. You have a very similar immobilization it seems. And then when we do the actual sample, you can see that in this case, in every other concentration except the zero concentration, we don't observe any binding. So in this case, in this particular molecule, we don't see any simultaneous binding. However, this is not yet completely evaluated. We have not really done any more examples. This is just a starting experiment. So we are thinking that we may need a possible control to really confirm that this assay is working for us, but the principle is quite straightforward. And also what we've done is evaluated single point mutations, just to evaluate what amino acids are important for binding. And here's an example showing you the original binding molecule. So on the left you have 3 panels showing different mobilization levels of the target, and on the right you have 2 surfaces with each same. And we compare that to our mutants. So as you can see, we have very nice binding to the target, and we have quite nice binding to each same. And you see the KD values are calculated, and they are quite similar between the surfaces here. Quite similar between the surfaces here as well, even though we have quite a low immobilization level. And here's an example from one of the mutants, and it's still binding the target very nicely, and we have comparable KD values, and the HSA values are also very similar, so this indicates that this mutant is still binding, not affected at all. And also, here's another example where you can clearly see that target binding is low. And this can also be observed if we would raise the concentration. And the HSA binding is intact. So it's very nice that in just one injection you can just get your answers immediately. So very, very quick process. So just to summarize, we have the ability to engineer bispecific single-domain proteins, as you've seen. And we can do faster a lot of kinetic analysis on these. We have integration of replicate sampling, which is very quick. You don't have to really worry about doing it over and over again. It's just take a day and then you're done. The format enables us to be creative. We can think of all kinds of ways to test your interactions. You can have multiple analytes, and you can do all kinds of tricky things. And it can be extremely time efficient in some applications. So that depends a little bit how you define it. So we find out a way to, like in this episode, the evaluation experiment which I showed you with the growth factor, it's a very time efficient. You get a lot of replicates quickly. If we would do the same thing if we would do one injection at a time, we would probably spend days or even weeks getting one of that. And also, just on the downside, there are some technical challenges to overcome. There's some trial and error involved, but that's always with research. So that's it. So I would like to thank you for your attention.